Hi. In this video, I want to do a hopefully quick refresher of the basics of genetics that you'll need to excel in this class. And here I'm focusing on Mendelian genetics. That is, we think of it as one trait usually determined by a single gene. Of course, that's different to what we're going to be discussing in quantitative genetics when we're talking about continuously variable traits that are usually determined by many different loci in the genome. So here is an example vocabulary list that I really hope you would be able to fill in by the end of this video. Let's start using some of those vocabulary terms talking about these famous genetic experiments with Mendel's peas. So Mendel had round and wrinkled peas in this parental generation. The words round and wrinkled describe the phenotype or the appearance of the trait. The round peas produce these round seeds, the wrinkled peas produce these wrinkled seeds. When Mendel crossed those peas, he found that the offspring were all round. It's important to note here that the parental generation of peas were true breeding. That is, when you crossed round to round or self to round, you only got round, and the same for wrinkled as well. Yet, when you cross them, you get round, the round phenotype in the F1 generation. So what we just saw was dominance. Dominance is the relationship between alleles, where one allele is going to express a phenotype that masks the phenotype of the other allele. We just saw that with the round and wrinkled peas. There was some genetic basis for the round phenotype that was dominant to or masked the phenotype of the allele that conferred the wrinkled phenotype. Notice here I'm talking about the relationship between alleles, not phenotypes. I know we have a tendency, just in kind of casual language, to say things like, oh, brown eyes are dominant to blue eyes, which is problematic for a number of reasons. But the one that is really important in this class right at this moment is because that's describing a relationship between phenotypes, not between alleles that determine a phenotype. I think that's easier to depict when we put the genotype, that is the genetic sequence that we're talking about here, onto this map. So what we were seeing in this true breeding round P is that they have the genotype big R, big R. I just chose these letters to represent that allele. The wrinkled peas have the genotype little r, little r. Again, I chose the letter R to represent this gene, and the little r denotes that that's a variant allele at this locus. So, if we're remembering how this is going to work, this P in the parental generation, big R, is only going to produce big R gametes. This P from the wrinkled parent is only going to produce little r gametes. The big R and little r gamete come together to form the offspring, which will be heterozygous. That is, it has one of each allele. And here we're seeing the dominance of the big R allele and the fact that even though the genotype is heterozygous, both alleles are present, you're only observing the phenotype that's associated with big R because it is completely dominant to the little r allele. So now what happens if we cross that F1 generation of heterozygous round peas? Here we'll go to a Punnett square. And now when you're using this, you're writing the haploid gamete genotypes on either side. So for example, I know that one of those parents is going to produce gametes that either contain the big R or the little r. Because it's a heterozygote, it has both alleles. So the haploid gametes will be either big R or little r. And so will the other one because it is also heterozygous. So now it's just a matter of filling in these possibilities. So one way that those gametes could come together is that both of the big R's could come together to form a big R offspring, or the offspring could inherit the big R allele from one parent and the little r allele from the other parent, or the reverse is true. They could be heterozygous but have inherited those alleles from the opposite parents. And then obviously the last combination is that the offspring could be little r, little r. So we know that we'll see the homozygous big R, the heterozygote you can achieve in two ways, depending on which parent you inherit which allele from, and the homozygous for the little r. And this ratio is what you would see in this situation with complete dominance of the big R allele. So 3 to 1 of all of the offspring would be in the round to wrinkled phenotype. This messy slide is just to demonstrate what is the genetic basis of an allele. When I draw that big R and little r, what do I mean? So what you're looking at here is an amino acid sequence alignment to one of the SBE1, these starch branching enzymes in peas that are responsible for this wrinkled and round phenotype. And so you'll notice that there's one sequence on the top of every line that is associated, you could think, with the big R allele. And then there's one on the bottom that is the little r allele. And this is an amino acid sequence alignment. So we're obviously talking about a protein coding region of a gene. Remember that not everything in the genome, not every locus in the genome is a gene at all. And not every region that we would say is within a gene is protein coding. There are regions of the gene that will never be translated to protein. And there are coding and non-coding and introns and exons and promoters and enhancers, like all of these different regulatory regions, and then just regions of the genome that are kind of junk that we don't know what it is, but it sometimes is associated with a trait. So that's something that you might want to refresh too. So as you're going along, you might see that these are looking really similar, the big R allele and the little r allele. There aren't that many differences until you get here to the end and you see that there's a gap in the alignment. So the little r allele is missing an entire section of this protein. It's missing it because there was an insertion that disrupted the sequence at this point in a way that basically caused a big deletion of the amino acid sequence. And that is enough to make that version of the allele completely defective. In the case of the starch, I cannot say that, starch branching enzyme that we were just talking about with the big R and little r alleles that cause the wrinkled and round phenotype in peas, that little r allele, the protein product that is translated from that little r allele is non-functional. So that's contributing to the fact that the other allele is completely dominant over it. But imagine a different sort of biological situation. Say, for example, that now we're talking about pigment production in flowers. And so maybe you have a gene, again, we'll call this locus R, and where big R is you produce lots of red pigment, and little r, you don't produce any pigment at all. But if you're heterozygous, then you still produce some pigment, and maybe that's enough to give you some sort of shade that's in between. So this might be a situation of incomplete dominance, or you're not totally recapitulating the phenotype of the homozygous big R in the heterozygous, but it's kind of something in between. Now, in quantitative genetics, we're going to be talking a lot about traits that are affected by more than one gene. And let's start kind of small and still think about a trait that undergoes Mendelian processes and the way that it manifests. Let's talk about a trait that's determined by two genes. There's this really famous example with Labradors that is very cute. I encourage you to revisit this. I'm actually going to talk about kind of simpler fictitious example that I find particularly illustrative. I just wanted to have pictures of dogs. So here's my silly example. Let's say that we have a pigment gene, so some locus that's associated with making pigment and hair color. So this gene has two alleles. You could be little p, which produces a light yellow pigment. You could be heterozygous, which would produce a red color, or you could be homozygous big P to produce a brown color of hair. Let's add another locus too. So now let's say that there's also a follicle gene, which we'll call F. And now if you are homozygous, little f, genetics is the only reason that I still remember any cursive, by the way, is to help me with these lowercase letters. Sorry for my handwriting. So little f would mean maybe that you do not produce any hair, you're totally bald. But the heterozygous condition or being homozygous big F, let's say that this is completely dominant and you have a full head of hair. So, what color hair does a person with the genotype big F, big P, little p have? Okay, so I put this little guide here for us. So the heterozygous for P means it's red, and then homozygous big F means a full head of hair. So they look like this. They have red hair, right? Yay. Okay, what color hair does a person with genotype little f, big P, little p have? Okay, so the Heterozygous P was red again. So this person has rut-row. Little f is no hair at all. So what color hair does they have? It's kind of a meaningless statement. That is to say, in genetic terms, that you have to have the follicle, the right follicle combination of follicle alleles in order to have hair at all. So if you don't have the phenotype that confers hair growth, then the gene that's determining the color of your hair is kind of, it doesn't matter. That is to say, the follicle gene is epistatic to the pigment gene because it kind of acts in an upstream process to the having hair at all. So the follicle gene is epistatic to, or the etymology of that word is kind of on top of and stopping. The follicle gene is masking the allele here. It doesn't matter what genotype this person has at the pigment locus, they're just bald. They don't have any hair at all. Now, to the last thing that I want to talk about is the concept of linkage, or can we get all combinations of alleles at all genes in the offspring, or are they equally abundant? And this depends on recombination and independent assortment, other terms that you might want to revisit from some previous genetics work. But I'm going to draw out three examples here. So each of these three circles are nuclei of diploid cells. These are diploid cells that have two chromosomes. You see there's the bigger one and there's the smaller one, and they're diploid, meaning that they have a copy of this longer chromosome and then a homologous copy of that chromosome. So they inherited this copy from one parent and this copy from another parent, and the same for the other chromosome that they have here. So can we get all combinations of the P and F alleles in the offspring of this diploid cell? Let's say that this is a sex cell that's going to form haploid gametes. So just based on the same properties that we saw in the Punnett square before, what I see here in this heterozygous individual is that depending on which chromosomes I randomly picked out of that cell, I could get big P, big F, big P, little p, big F, or little p, little f. And I'm going to get those in equal proportions because they're completely unlinked. They're on separate chromosomes. And the law of independent assortment means that those chromosomes are going to be randomly sorted into the gametes that are formed by this sex cell. Now let's think about a situation where the P and F loci are on the same chromosome, but really far apart. Here I've drawn them on opposite ends of this larger chromosome. So if you were just to select those chromosomes right now as they are, it would be possible to have a gamete with the big P, big F genotype, right? Because let me get my laser pointer back. I could grab this chromosome right here, and I could grab this chromosome right here. So if I grab this one, I get big P, big F, and if I grab this one, I get little p, little f. But so let me draw that one too. So I get the little p, little f. Now, is it possible for me to get these other combinations of alleles? And the answer is yes, through recombination. So if there was crossing over between homologous chromosomes at any of these locations between the P and the F locus, then that is how you could get the recombinant genotype. Or in this case, that would be the big P, little f, or the little p, big F. Now, if these loci are really, really, really far apart, and recombination is just definitely going to happen between them, then maybe we could say that we're kind of maybe approaching equal proportions of those. But you might expect to see a little bit more of the big P, big F, and little p, little f. That is the concept of recombination distances and linkage mapping. The frequency with which you observe recombination between two loci on the same chromosome is directly related to the distance between them. Okay, so here's our last scenario, is that the P and the F loci are on the same chromosome and they are really, really, really close together, super close together. So what combinations of alleles are we going to see in the gametes formed by this cell? Well, so again, I can get this chromosome going segregating into a haploid cell, and this chromosome segregating to the haploid cell. So I know that I'm going to see big F, big P, big F, and little p, little f. Um, but now, in order for me to get the recombinant genotype, there's only one place that recombination can happen, and it's so tiny that like I can't even draw it. In other words, it's very rarely, it's not going to happen very often. It's going to be a really rare event. Most likely, recombination will happen between these homologous chromosomes somewhere else, somewhere outside of the distance between those two loci, because most of the chromosome is outside of the distance between those two loci. It's just a probability situation. So you may observe the big P, little f. Sorry, my throat is going wonky. Little p, big F, but they are going to be extremely, extremely rare. So again, if we're expecting to see combinations of all alleles at two loci, it depends on their linkage state. That is, are they on totally separate chromosomes, completely unlinked? If they are on the same chromosome, what is the genetic distance between them? That is, how frequently is recombination going to occur to give us that recombinant genotype? I think that's all I have. That was an entire semester of genetics in one video. So I hope that helps. Thanks.